Thank you very much for the introduction and the invitation. And in this talk, I want to introduce you this concept, topological landscape, which I found pretty useful for elucidating the living matter and its structure that we encountered. Specifically, today's story is about how we map a living matter on the left to a living landscape on the right, which gave us a new interesting intuitions about the system. And of course, this work was possible because of the amazing team from a combination of Fakhri Lab, Dunkel Lab, and Skinner Lab, and also these funding sources. So, biological forms often emerge from living collectives, tissues from cells, biofilms from microbes, and flux from birds. And importantly, these forms are not programmed top-down, but are self-organized through interactions between these living entities. And also, these self-organized structures are often dynamic, constantly changing, and can be far richer than non-living counterparts, as a wider possibility of interactions are allowed. In particular, for this project, we are interested in type of interaction which is called non-reciprocal. So what do you mean by this? Newton's third law states that action and reaction is equal and opposite, which is always true at microscopic scales. And in fact, in equilibrium, this action-reaction symmetry holds even for effective interactions mediated through the environment. Nevertheless, all the living systems are fundamentally non-equilibrium, and their action and reaction now no longer needs to be symmetric. Like in this video, how a hare will effectively pull the wolf to be closer, while the wolf will push the hare to run away. So these kind of non-reciprocal interactions are particularly relevant for living systems. But and our question is how this non-reciprocity reshape the self-organized structures. So this is our approach. Our lab uses experimental system of interacting embryos to study how living systems can self-organize structures depending on their underlying interactions. And before the video runs, I'll give you the sense of the scales. We are basically looking on the top view of a 6-well plate filled with seawater. And this system is few millimeter scale, much larger than the individual starfish embryos shown here. And we will progress over hours, and the embryos will appear in this field of view and self-organize their structures for hours. And let's see. As you can see here, the embryos form a remarkably ordered crystal structure that also self-sustains a chiral dynamics. It rotates by spontaneously for days, which is why we call this a living chiral crystal. So what kind of interactions underlie this self-organized structure, and how will we change this? Let's zoom in. On the left, we see a side view of single starfish embryo 24 hours after fertilization. And this starfish embryo in nature would need to survive in the ocean, basically. And it turns out to be a good swimmer. With they have the cilia in the surface that conduce the continuous stroke, which generates hydrodynamic flow. And this allows them to push the fluid downward and arise to this air-water interface. Now, from the top view, we see that these embryos are also chiral, rotating clockwise due to the chiral symmetry breaking in its activity. Moreover, we can see, if you look closely with these PIV particles, we can see that the embryo is actually sucking up these small particles, which means that it is sucking up the fluid on the plane to push it downward. And this inward flow leads to an effective long-range attraction between a pair of embryos so that they form a stable bound pair here. In addition, the chiral rotation of embryos make the pair rotate as well. And this kind of pairwise interaction is the one that was underlying the self-organized crystal we saw in the previous slide. Right? So how did this platform allow us to study non-reciprocity? Building up the same embryo pair, we tune their interaction by introducing developmental gap. The embryo in the left, E1, is the one that we used previously. And the embryo on the right, E2, is older than E1 by 24 hours. And so it's larger, more elongated, and generates quantitatively different hydrodynamic flow from E1. As a result, as we see in this top view video, the embryo pair exhibits a striking run and chase dynamics. E1 is making E2 to run away, while E2 is making E1 to follow. And this run and chase dynamics, which is mediated by the hydrodynamic flow in the media, highlights how the interaction between these two embryos are non-reciprocal. Now, let's see how such non-reciprocal E1 E2 pair realizes a different self-organized structure. Again, we see a top view of a six-well plate filled with seawater and thousands of starfish embryos just ready to come up to this air-water interface. Some of them already came out. Since now we have two different types of embryos, we now mark E1 as blue and E2 as red, overlaying their short time trajectories on the video. So let's see the first 3 hour dynamics. Here, it's very different from what we just saw earlier. Instead of forming a single crystal, the embryos are forming multiple clusters. Each individual cluster is quite dynamic, and as in the pair case, the E2 region is kind of leading the E1 region. And these clusters can transiently bound to form a larger one, but due to the non-reciprocal interaction, they are not really stable and breaks apart. And this is how the structure is sustained its own dynamics for hours. Now, however, around 5 hours after the experiment onset, we see that the self-organized structures has transitioned. Instead of forming multiple clusters, now the embryos form a single-ish cluster, and instead of traveling far away, each embryo, from the trajectory, we can see that each embryo just jiggles around near its lattice position. We thought that this different behavior and the emergent self-organized structure can happen because our non-reciprocal interaction can change over time as these embryos develop during the experiment. And using a recently developed interaction inference technique, we quantify the effective pairwise interactions between the embryos, which consists of the long-range hydrodynamic attraction and the short-range steric repulsion. And as we do this inference based on the embryo trajectories over this early time window, but also over the late time window, we immediately see that the development over time in our experiment makes difference, brings the E1 embryo to be more similar to E2 embryos as it ages. And in fact, this time dependence is very nice because it allows us to study not just a single self-organized structure, but the whole self-organizing process driven by the transition in the interactions between the individuals, which is often the case in even the natural self-organizations, like in the developmental process. So basically, we have observed non-reciprocal self-organization, which undergoes a transition from a more dynamic multi-cluster case in the case of strong non-reciprocity to a stable single cluster case in the case of weak non-reciprocity. And this is exciting because now we have a self-organizing process in the lab that is quite tractable with simple microscopy. And we are left with one challenge. How can you possibly characterize this self-organization? The snapshots definitely look quite different in eyes, but they are both similarly complex and far away from a perfect crystal, and we need some measure to actually quantitatively assess their differences. So this is how what made us to develop a new toolkit, which is what we call topological landscape. What inspired us is the concept of topological transitions, especially the ones called T1 transitions, which has been already used in self-organized structures such as tissue dynamics and morphogenesis and many others. So let's imagine that we are zooming into monolayer cells, A, B, C, and D here. And here, initially, C and D are the nearest neighbors. And if we imagine this imaginary microscope, we'll see that each cell's position will fluctuate, like jiggle around, but such small fluctuation will usually not make a radical change, and each cell will basically keep their neighbors. But then, if you inject sufficient energy, then you may transition from this A panel to B panel. The system may deform enough so that you can pass through this what a configuration in B and settle down into a new configuration where now A and B are the nearest neighbors. And if you make this notion of the transition a little bit more abstract, is a transition in the graph of the nearest neighbor network, like the one, especially the ones that marked red here. And this graph can be a network of cells, but also network of anything, like the embryos. And that's how we map our embryos into a neighbor connection network, like here. In particular, for each embryo, we characterize its next to nearest neighborhood as what we call topological motif. For this embryo in the bottom right, its local neighborhood corresponds to a perfect crystal with all the nearest neighbors and itself having a six neighbors. On the other hand, for another embryo, its neighborhood may not be that symmetric, going away from such crystalline state. Now, the whole spatial structure in each snapshot becomes a distribution of topological motifs. But at this point, the distribution itself does not really give much intuition of what the key feature of our system is. And basically, we need a final step to better understand this distribution on the right, which is a distribution of topological motifs. What we do is that we leverage the fact that these motifs have a pairwise distance in terms of the number of the T1 transitions, the topological transitions we just saw earlier between them, which will in physical space probably incur some energy cost. So this has we need some sense of the physical distance, physical like energy barrier is distance. And using standard dimensional reduction technique, we then assign a fixed low dimensional coordinate for each motif. And this will basically put the topological motifs who are close to each other, also close together in terms of these coordinates. So yes, this is the low dimensional manifold. And finally, we use this frequency motifs as the weight of the embedded space, converting the structures into a landscape of local network topologies. In particular, like in this topological map, the landscape can now have peaks. Peaks, okay. On the like in the top right, top left of the topological map, and these peaks of the topological landscape will basically let us know what is the most probable configurations in the system. So let's see the topological landscape of our experiment over time. We can really see that the landscape has two peaks. And by marking the most dominant 14 motifs from the experiment on this manifold in the left, we can find that each peak corresponds to the two most dominant motifs. This peak on the right corresponds to a perfect crystal, which we call M1. And another peak on the lower left corresponds to almost crystal, but with one nearest neighbor having not a six neighbor, but five neighbors. And this is what we call M2. Now, over time, as we can see further in the video, this subordinate structure transitions to a more crystalline structure. And this is also reflected in the landscape as the M1 peak is growing stronger over time. And we can see this even more quantitatively if we focus on this M1, M2 axis and slicing our landscape, which slices our landscape through it. And that's how we finally arrive at this diagram. We are basically plotting the negative log likelihood, which can be intuitively thought as some sort of energy landscape, as it means that the system likes to be in the bottom of the well. And this is what happens initially. Initially, like in this perfect curve, we can see that in our system, the M2, which is not the perfect crystal, is the most likely configuration. But as time goes, there is a time point where M1 and M2 are equally likely. And finally, later, the system keeps going and M1 becomes more likely than M2. And this analysis was very useful for us in two ways. First, it clearly shows that our system is essentially transitioning from a M2 dominated state to a M1 dominated state. Moreover, the way transitions, which is through a coexistence state, brings the analogy of something like water turning into ice, or technically a first order transition. While this is only analogy, it provides a powerful intuition that we did not really have when we just had our experimental video. And this intuition also led us to new testable hypotheses, which turned out to be true and made our understanding of the system even deeper. So now we are at the takeaway of this talk. First, we used we had the experiment with the self-organization process with tunable interactions between the starfish embryos. And next, we developed a topological tool for understanding these complex spatial structures. And finally, this topological tool um, provided us the intuitive picture for how the structure of this starfish embryo system transitions. And I now noticed that I was too conservative on my timing, so my talk was only covering basically figure 1 and figure 3 of my work, and there are many other open avenues, and they're left, let's see, mm, beyond these slides. First of them is the model simulation. Since we were inferring the interactions from the experiment, we could also modulate the interactions of as a particle between particles in the simulations based on the inferred interaction. And this interaction in particular could be systemically altered to have a shift from fully reciprocal case to a more strongly non-reciprocal case, even stronger than what we see in the experiment. And as a result, we systemically assess how the self-organized structure transitions as we tune the non-reciprocity in the interaction. Another thing we did was to develop a topological, what we call topological flowscape, which is basically maps how a landscape, the trajectory of landscapes are quantitatively distinct from one another in terms of their trajectory over the self-organization. And in particular, the axis in this topological lens flowscape we see is of information measure, and we are curious of how this can further um, lead to a more direct connection between the information theory and non-equilibrium thermodynamics to this self-organization process. And of course, we can also think of other systems like epithelial tissues or many other systems which are definitely not like super ordered, but also not random and trying to understand the underlying system, underlying structures. And with this, I'd like to end my talk, and I guess I'll have plenty of time for questions and looking forward to it. Thank you.